So in this video, we're going to be talking about photosynthesis, or how plants use sunlight to make energy. Now, if you remember the formula for photosynthesis from your high school biology class, you'll remember that 6 carbon dioxide molecules and 6 water molecules come together to make a sugar, more specifically glucose, and 6 oxygen molecules. And the energy for that is provided by light. So in order to take light, which is a pretty useless form of energy, and turn it into usable energy, plants have a couple things that animal cells don't have. So we have our plant cell here with a vacuole in blue and a nucleus, a cell membrane, and a cell wall. It has mitochondria just like animal cells, but it also has these organelles called chloroplasts, which are a lot like mitochondria. They have 2 membranes, they are very intimately involved in energy production, and they're going to be the site of photosynthesis. So let's talk a little bit more about the chloroplast. Now, the first thing you're going to realize is that chloroplasts are a lot like mitochondria. They have 2 membrane layers, they have DNA, and they use that DNA to make their own proteins. And in order to make their own proteins, they have to have ribosomes, which I forgot to draw in here. So we're going to add some ribosomes. And just like mitochondria, the fact that they have their own DNA and they make their own proteins supports the theory of endosymbiosis, or that chloroplasts were once their own cells, their own bacteria that got engulfed by a larger cell and then began to live in it. And they're also going to have some other things that are specific to plants, like starch. But what we're really concerned about are these pancakes. Of course, they're not actually called pancakes. The real name for these structures are thylakoids, and the thylakoids are the site of light-dependent photosynthesis. So each of these little individual pancakes is a thylakoid, and a stack of pancakes is called a granum. Now note that each thylakoid in a granum is connected. They're sharing the same cytoplasm. That's just something to make note of. So we've talked about what's inside of a chloroplast, but we haven't actually named the compartments. So the inside of the thylakoids is easily named the thylakoid lumen, and the inside of the actual chloroplast, the site where all the DNA, the ribosomes, the starches, that's called the stroma. And different parts of photosynthesis are going to take place inside of the thylakoid lumen, across the thylakoid membrane, and inside of the stroma. So what about protons? You'll remember that protons are really important for energy production in the mitochondria, but their location inside of chloroplasts is a little different. Inside of your thylakoid lumen, we have lots of protons. But on the outside of your thylakoid lumen, in the stroma, we don't have a lot of protons. We have few protons. And we're going to talk about why that is right now. So first we're going to introduce just the overall reactants and products of photosynthesis. So on our left here we have a mixing bowl that represents our light-dependent cycles. I thought mixing bowls would be appropriate since they make cookies and sugar is our end product. And on our right here we have our Calvin cycle. Now the first thing that we're going to put in our light-dependent cycle is obviously light. Hold on, I don't like the way I drew that arrow. So we're going to have a nice big lightning bolt coming in here to represent our light. Now notice that I'm drawing an open-headed arrow. Open-headed arrows will be molecules or sources of energy management. Closed arrows will be sources of substrates. And then we have our first substrate, and that is water. So you'll remember the formula is 6 CO2, 6 H2O. So this is where our water comes in our light-dependent cycle. And then we have our energy management molecules. So the first energy management molecule is NADPH. So we're going to draw this in here. Notice that I'm saying NADPH, not NADH. In respiration, NADH is used as an electron transporter, but in photosynthesis, it's NADPH. So what the light-dependent cycle is going to do, oh my gosh, I made my own mistake. I drew NADH. Let me fix that. So what the light-dependent cycle is going to do is reduce that NADP to NADPH. And next we're, of course, going to produce ATP. So you can see right away that this light-dependent cycle is going to be producing all of our high-energy molecules, our ATP, our NADPH, and all of that energy is going to be used to power the formation of longer carbohydrates in the Calvin cycle. So the first products of the Calvin cycle are going to be ADP and NADP+, the oxidized forms of those energy carriers. But we're also going to need substrates. We're going to need something to make those carbohydrates with. And that's going to be CO2 and then a sugar called ribulose bisphosphate, which we're going to abbreviate here as RUBP. RUBP. So that's ribulose bisphosphate. And those sugars are going to be used to make G3P, glyceraldehyde 3-phosphate, which you might remember is an intermediate in glycolysis. So now we're ready to look at the details in each cycle, and we're going to start with the light-dependent cycle. This is going to happen in the thylakoid membrane. So we have our thylakoid membrane right here in blue. And that means that on the bottom here, we have our thylakoid lumen. So remember, your thylakoid lumen is the really, really proton-rich area. And then on the top, we have our proton pore stroma. And that's going to be important because the light-dependent cycle is a lot like the electron transport chain. So these proteins are going to be used to establish that proton gradient. And the first protein is photosystem 2, which is followed by plastoquinone, which we've abbreviated PQ. And after PQ comes cytochrome B6F. And B6F is going to be followed by this little guy I forgot to put a face on, plastocyanin. I remember plastoquinone and plastocyanin because they start with plasto and people make plastic plants. Just my tip. And after plastocyanin, we have photosystem 1. So remember, photosystem 1 comes after photosystem 2. And then on top of photosystem 1, sort of working alongside photosystem 1, we have ferredoxin. And, of course, ferredoxin NADP reductase, or FNR. And our last little guy here, possibly the most important part, well actually they're all pretty important, is ATP synthase. And he's going to work just like the ATP synthase in our electron transport chain. Okay, so now that we know what everyone's name is, we can look at the actual function. And the first part of the light-dependent cycle is of course going to be the light that strikes photosystem 2. And that's going to give photosystem 2 the energy it needs to take water and break it down, so take some water here, and we're going to break that down into O2 and H2O. So notice we're producing protons, which are going to help contribute to that proton gradient that we need to make ATP. And when we take that water and we use light to break it down, it releases electrons, and those electrons are going to be passed to plastoquinone. Just a quick note, just like in the electron transport chain, electrons are passed from the least electronegative to the most electronegative molecule. So those electrons on plastoquinone are going to be used to pump protons to B6F and then into the thylakoid lumen. And then the electrons will be passed on to cytochrome B6F, and from there onto plastocyanin. Remember, each subsequent protein is going to be a little more electronegative. And finally, they're going to end up on photosystem 1. And photosystem 1 can also use light energy, but it's not going to use it for as explicit of a process as breaking down water. And those electrons will then be passed to ferredoxin and ferredoxin NADP reductase, which is going to reduce NADP, just like its name suggests. So we have some NADP+, right here. Remember, that's what we talked about. That was one of those initial ingredients we needed. and it's going to produce NADPH, which is going to be really important for making those longer carbon chains in the Calvin cycle. But we also have this proton gradient here. And this proton gradient, so we have a ton of protons in the thylakoid lumen, which is going to mean we have a really low pH in the thylakoid lumen. Remember, more protons means a lower pH. And those protons are going to want to go to the stroma because there's not a lot of protons in the stroma. And that movement of protons through ATP synthase is going to power the formation of ATP from ADP. So we have our adenosine diphosphate, ADP, and ATP synthase is just going to combine that with a phosphate, making high energy ATP. So now we're going to be talking about the Calvin cycle, which, as you'll remember, will happen in the stroma. So there's 3 parts of the Calvin cycle, and I'm going to try and draw them all in different colors. And the first part is where we take all those carbon substrates and we combine them into a longer chain molecule, which is eventually going to be cleaved in half. So we start with our ribulose bisphosphate. You'll remember that was one of our substrates that we added to our Calvin cycle mixing bowl. And that's going to be combined with CO2. I don't want to draw our oxygen as a carbon to confuse you, so our CO2. So essentially what's going to happen is one of those carbon molecules is going to be added onto the end of ribulose bisphosphate, and that's going to make a 6-carbon molecule. So we have our 6 carbons here, 1, 2, 3, 4, 5, and 6, and that is going to be broken down into 2 3-carbon molecules called 3-phosphoglycerate. Now, even though those are 3-carbon molecules, just like glyceraldehyde 3-phosphate, they have a slightly different structure. Alright, and the only enzyme that I'm going to talk about is rubisco, which is responsible for this whole process, the whole carbon fixation phase. You're fixing one carbon from CO2 onto ribulose bisphosphate. Now, our next phase is reduction. So all reduction is going to do is take those high energy molecules, so ATP and NADPH that we made in the light-dependent cycle, and oxidize those so that it can reduce our 3-phosphoglycerate. So we oxidize ATP, oxidize NADPH, and we reduce our 3-phosphoglycerate to glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate, you'll remember, is a component of glycolysis. So there's all kinds of things that the plant cells can use that for now that we have it. But the primary thing it's going to be used for is the regeneration of ribulose, or the regeneration phase, which I'm going to draw in orange here. So most of our glyceraldehyde 3-phosphate is going to be funneled right back into this cycle. And in order to regenerate that ribulose bisphosphate, we're going to take 1 ATP and hydrolyze it. Of course, not all of it will be used for regeneration. Some of it will be released so that it can be used in other cellular processes, but most is used in regeneration. And that's it for photosynthesis. Bye!